Greetings, everyone. Today we're going to look at the nervous system. We're going to look at the physiology of the nervous system as it relates to pharmacological principles. So lots of big words to say, nervous system and drugs and how the two interact. Important for us to know this because the majority of the drugs that we give in the out-of-hospital setting directly target some component of the nervous system to do something to the blood pressure, to the heart rate, generally speaking. So let's take a look at this. This is really important stuff. Again, I apologize for the banners. I'll get rid of that as soon as I can. All right, so this is just a little refresher slide, and the refresher slide is, wow, overwhelming, right? Overwhelming stuff. So this is a picture of the sympathetic nervous system. The sympathetic nervous system is also called the thoracolumbar division of the nervous system. Thoraco from the thorax, and lumbar, of course, lumbar. So this is the thoracolumbar division of the nervous system. The sympathetic nervous system, as you can see, the nerve fibers originate from the thorax all the way through to about the mid portion of the lumbar spine, and that's where all these fibers originate. That's where they initiate. You also notice a couple other things here. You'll notice all of the end organs, the final organs destination of the nervous system, and you should be able to look through this and be able to determine what exactly the sympathetic portion has to do to each of these end organs. So for example, the eye, you would want to be able to say that you have pupillary dilation. All right, remember, this is fight or flight, so when it's low light conditions, we need to be able to dilate the eyeballs, and the sympathetic nervous system does that. Salivary glands, they decrease their secretion of saliva because we're not trying to digest anything at that point while we're trying to run away and save ourselves. Heart, a couple things are going to happen. Increase in rate, increase in force of contraction, all right, and also increase in how quickly conduction takes place. So increase in conduction velocity. So this means this is the heart rate itself, so it'll go from heart rate of 80 to a heart rate of 180. The force means that it will be a more forceful contraction. And then the conduction velocity, this has to do with the conduction system and the speed at which nervous system impulses conduct through the heart to make it contract faster. All right, so we'll explore these in a lot greater detail. I just want you to be able to associate sympathetic nervous system with these changes. All right, the lungs, if you're running away, this is bronchodilation. This is so we can get more air in other words, so we can get more oxygen in and more CO2 out. So the liver, gallbladder are going to do a couple things. Liver is going to release glycogen stores so that we can increase glucose in blood plasma. Stomach's going to stop working for now, so we're going to have a decrease here. So we're going to take a look at large intestine and small intestine and just say decrease blood flow here. And the reason for that is because it wouldn't be necessary to do anything to this at this point since we're trying to run away. Adrenal medulla, we're going to look at this in a few slides. We're going to increase norepinephrine release here. This is going to be one of the ways that we chemically dump norepinephrine into the system. All right, kidneys are going to shut down in terms of urine production, and the reason for that is we're going to try to maintain all of our fluid so that we can maintain blood pressure. We'll get to the gonads a little bit later. This is the important stuff I want you to know for the sympathetic or the thoracolumbar division of the nervous system. So thorax, lumbar, that's where these fibers originate. All right, so there are a couple other important pieces here to note just for now. And I don't want you to spend a lot of time worrying about this stuff. I'm just going to point it out as a piece of trivia right now. I want you to associate, we're going to see this in greater depth here in a few slides, but I want you to appreciate that as the nerve leaves the spine, it connects to this bundle here, and this bundle is called a single ganglion. So a ganglion is a collection of nerves, a bundle of nerve connections that exists. The plural of this is ganglia. All right, ganglia. So ganglia is the plural, and ganglion is the singular form. Important to speak correctly. So you'll notice that that the fibers that leave the spine and get to the ganglia, they're pretty short. they're pretty short compared to after the ganglionic terminal here. You'll see that these nerve fibers are really, really long, right? They go all the way over to the target or the end organ. So it's important to remember that the sympathetic nervous system has a couple things. So the ganglion connections, where the connections are made at the ganglia, these are called the synaptic area. There are a bunch of synapses, there are a bunch of connections that take place in here. So preganglionic, everything before the ganglia, in the sympathetic nervous system, preganglia is a short connection. So preganglionic, short fibers. Postganglionic, after, long fibers. Right? So that's kind of an important thing here. The other thing that you'll need to know here is that for every single one preganglionic fiber that exists, there are about 20 postganglionic fibers that depart. So, sympathetic nervous system, preganglionic, that's this area here, preganglionic, these are short fibers. Then you have the ganglion, and the ganglion makes all the connections between the pre- and post-ganglionic fibers. In the sympathetic nervous system, post-ganglionic fiber is a long fiber, and in addition to that, for every single pre-ganglionic fiber that exists, there are about 20 that leave each ganglion from on the other end there, from the post-ganglionic end. So this is going to play an important role later, because sometimes we're going to target divisions inside the ganglionic area. Sometimes we're going to go straight to the end organ with our drug to affect a change. And this is kind of important. We're going to talk about this in just a second, about the somatic portion of it, the things that control the skeletal muscle. And you'll see that in the parasympathetic nervous system and in the somatic nervous system, we have a 1-to-1 ratio. So for every single preganglionic fiber, there is exactly one postganglionic fiber. And really, in the sympathetic nervous system, this speaks to the complexity and the interdependence of the systems in the sympathetic nervous system. That is to say, where you have one preganglionic fiber that sends out a stimulus, it may actually be talking to 20 or 40 or 1,000 other little components inside the body. And that speaks to the collaboration that takes place between body systems to maintain and to regulate all of the things that it does. All right, so that's probably a lot of information. I don't want you to worry about that. You can certainly re-listen to this. You can read your text. There are a tremendous amount of resources online to help you figure this out as well. YouTube's another great source. But I wanted to introduce it to you for now. All right, so the next component here, this is the parasympathetic nervous system. And parasympathetic nervous system is also known as the craniosacral. And craniosacral because the nerve fibers for the parasympathetic nervous system, they originate at the level of the cranium and the sacrum. So craniosacral is parasympathetic. This is the feeder breed component. And we'll talk a lot more about the cranial nerves, right? So cranio, there are 12 cranial nerves, and we'll get to know these quite well throughout the course here. We'll see them quite a bit. And then there's the sacral component, or the sacral innervations as well. And as I mentioned to you in the previous slide, here again there are ganglia. Right? These guys here, these are the collections of nerve fibers that terminate on one end and originate on the other end. They communicate to one another. And there are a couple things that happen here. Well, first of all, in the parasympathetic nervous system, there is a 1-to-1 ratio of pre to post ganglionic fibers. So as you see here, if you look at, you take one nerve fiber and it gets to a terminal ganglion, and from there only one fiber leaves. Right? So this is a 1-to-1, and so the parasympathetic nervous system is going to very specifically talk to one thing with one fiber. So one thing being an organ or an organ system or a cell. All right, the other thing you'll notice here is that pre-ganglionic fibers are long. They're long. They originate here. They go all the way over here to the ganglia or a plexus. Preganglionic fibers are long, and the postganglionic fibers are short. So this is exactly the opposite of the sympathetic nervous system. In the parasympathetic nervous system, long preganglionic fibers, short postganglionic fibers. All right, so what else do we need to talk about here? We said craniosacral. We said they originate cranial and sacral origins of the spine and the head. And we talked about a 1-to-1 ratio. We talked about ganglia, long on the preganglionic end, short on the postganglionic fiber end. And just as we did on the sympathetic slide, you should also be able to figure out what we're going to do, what the effect is going to be on each organ system. So the eyes, they're going to pupillary constriction that takes place. Salivary glands, they're going to increase saliva secretion. All right, heart rate is going to decrease. Force of contraction, decrease conduction velocity. Whoops. All right, lungs, we're going to have relaxation, so bronchoconstriction, so to speak. All right, and then all these other things here. Stomach secretions are going to increase. Large intestine blood flow increase. Small intestine increase in blood flow. And that's so we can feed or breed. All right, and same thing with the kidneys. We can resume making normal urine again, and so on. All right, cool. All right, so I think that's all the important stuff here. So let's take a look at the next piece of this. And this looks like a busy slide, but this will all come together, I promise you, at some point. All right, as we talked about before, there were a couple divisions of the nervous system. One of them was the somatic division. That's this guy here. Then we talked about the autonomic nervous system. Autonomic nervous system here. And we also talked about the autonomic nervous system being broken down further into the parasympathetic and sympathetic nervous systems. Now, we need to take a look at a couple different things here. We need to look at how what molecules are involved with all this, and we need to look at how we're going to get an impulse all the way from the brain or from one part of the body all the way to an organ system or an organ. So in the somatic nervous system, as you'll remember, we talked about the somatic nervous system being directly connected to voluntary skeletal muscle, and you'll see that's the case here. And we're going to talk about the neurotransmitters again because these are really important, but you'll see that acetylcholine is the primary neurotransmitter that's responsible for converting the electrical signal that is propagated or that's conducted all the way down this axon. And when it gets to this little end area here, which is a synapse, we'll talk more about that later, you'll see that it releases a neurotransmitter. That neurotransmitter is called acetylcholine. Acetylcholine travels over to the neuromuscular junction, and it causes skeletal muscle contraction. And you'll notice that one of the big differences between the somatic nervous system and the autonomic nervous system is that in the somatic nervous system, there is only one single neuron that communicates from central nervous system all the way to the effector organ. That is not the case in the sympathetic and parasympathetic nervous systems. Here we have two neurons, and we talked about them already. We talked about them as the preganglionic and postganglionic fibers that originate in those systems. So this does a couple things for us. This allows for speed to take place. If you have a single system, it's directly connected. You get an impulse that's triggered from this. It's propagated down the axon. It gets to the synapse. It is chemically converted by the way of acetylcholine, which is the chemical conversion. It chemically stimulates skeletal muscle contraction, and boom, you get skeletal muscle to contract. So this is a very fast system. We're going to talk about myelin in a little bit. Don't worry about that for now. All right, next we want to look at sympathetic nervous system, and you'll see that sympathetic nervous system, short preganglionic fibers, long postganglionic fibers, and you'll see that at the level of the ganglion, there is also a conversion that takes place. So a nerve trigger is received by the body of by the nerve cell, and it is propagated in the form of an electrical signal all the way till it gets to this stimulus here. I should probably be using a different color so you can see what I'm doing. It gets to the synapse here, to this ganglion area, and here the electrical signal that was traveling down the axon is then converted to a chemical signal. And this is a gatekeeping mechanism. It's a regulatory mechanism. And in this case, in the sympathetic nervous system and in the parasympathetic nervous system, the ganglionic, the preganglionic neurotransmitter is acetylcholine. All right, so we have acetylcholine that chemically converts this electrical signal. It then sends it over and triggers a signal in the post-ganglionic terminal to be initiated, and that is propagated down this post-ganglionic axon. And you'll see that there are multiple points of delivery here. And in the sympathetic nervous system, once this gets to the terminal portion of this post-ganglionic axon, that impulse, the electrical impulse, is then again converted to a chemical impulse. And in this case, in the sympathetic nervous system, it's converted by way of norepinephrine. So this is the neurotransmitter. Norepinephrine is the neurotransmitter in the sympathetic nervous system, post-ganglionic, that actually causes a change in the effector organ. So it's going to impact blood pressure this way by way of norepinephrine release in the peripheral vasculature. It's going to increase heart rate this way. It's going to do all the things that it does by way of norepinephrine. All right, so I also want to point out the fact that the sympathetic nervous system also has this direct connect to the adrenal glands, which sit on top of the kidneys, and the adrenal glands, adrenaline, release adrenaline. So you get a trigger from the central nervous system, it's propagated down the axon, it gets to the adrenal medulla, and it tells the adrenal medulla by way of chemical conversion, the impulse is converted to acetylcholine. Acetylcholine gets to the adrenal medulla, and boom, that stimulates the release of epi and norepinephrine into the bloodstream. So this is kind of a cool system. When you get scared, if somebody scares you, you're almost in an accident, and your heart rate just jumps up and everything. You start getting sweaty. You're nervous. This is one of the things that happens. The sympathetic nervous system, central nervous system, sends out a message directly linked to the adrenal medulla. It is converted to a chemical message. It's a messenger. Acetylcholine chemically triggers the release of epinephrine and norepinephrine at the adrenal medulla into the blood vessels. And this then goes to all the different end organs, and it has an effect, right? Usually a stimulatory effect. All right, so what do you need to know so far? Sympathetic nervous system. At the level of the ganglion, it is acetylcholine that's the neurotransmitter. At the end organ, or the effector organ, it is norepinephrine that is the neurotransmitter in the sympathetic nervous system. All right, now let's take a look at the parasympathetic nervous system. Again, you'll notice parasympathetic nervous system has a really long preganglionic fiber, a really short postganglionic fiber. And you'll notice same thing here. When the cell body receives a message from one of these little guys here, it triggers an impulse, and that impulse is propagated down this axon, and it reaches the ganglionic area. When it does so, that electrical message is converted into a chemical messenger. In this case, it's acetylcholine, all of the ganglionic neurotransmitters, sympathetic and parasympathetic, acetylcholine, and it converts that message to acetylcholine. That acetylcholine travels across the ganglion to other cell bodies of other nerves. There, it triggers an electrical impulse. That electrical impulse is propagated once again down the postganglionic axon, and once it reaches the synaptic area, it then releases that electrical, or it's converted from an electrical signal back to a chemical messenger system. In this case, the postganglionic neurotransmitter of the parasympathetic nervous system is acetylcholine. All right, so acetylcholine then goes to the effector organ, and it causes a change to take place. All right, so that's a lot of information, and I appreciate that. Let's see if we can summarize that stuff. I do want to add one more thing, and that is also the dopamine receptor, because you'll see that again. So this is again a sympathetic nervous system. So sympathetic, and we have a short preganglionic. It gets to a ganglion. That ganglion then converts that message back to an electrical signal, and it gets to the level of the renal vasculature, and there it is converted into another chemical messenger, and that chemical messenger is dopamine. So dopaminergic receptors are located at the level of the kidneys, and they impact smooth muscle changes to the renal vasculature. So we'll explore dopamine a little bit more. I just want you to appreciate that in the sympathetic nervous system, there's this additional guy, and that's dopamine. It's a dopamine receptor. All right, so let's move forward and see if we can make sense of all this mess, because this is quite a mess. All right, so let's take a look at receptors, the neurotransmitters, and their end effects. All right, and I think this will help kind of straighten things out a little bit. All right, so let's start out by looking at parasympathetic. So if you look at the parasympathetic nervous system, okay, the neurotransmitter of choice here in the parasympathetic nervous system is acetylcholine. Right? Acetylcholine. So I want you to think about that. And anytime you see the term cholinergic, I want you to think about the parasympathetic nervous system. So acetylcholine, cholinergic, or choline, I want you to think parasympathetic nervous system. Since that's the primary neurotransmitter of the parasympathetic nervous system, think of those things. All right, so in the parasympathetic nervous system, there are two types of cholinergic receptors, and those two types are called muscarinic. Whoops. Let me spell that correctly so you can see it. Muscarinic receptors and nicotinic receptors. Nicotinic receptors. All right, so muscarine is a chemical, nicotine is a chemical, and when muscarine affects a different cell, it must mean that there's a receptor or something to receive that chemical, and so muscarinic receptors are areas of the cells that are able to bind to this chemical called muscarine. Nicotinic receptors are the same thing, except they bind nicotine. Everybody knows what nicotine is. So parasympathetic nervous system, neurotransmission completed and accomplished by acetylcholine, and I want you to think that acetylcholine has to do with cholinergic receptor sites. Cholinergic receptor sites are further broken down into two main types. One is muscarinic, one is nicotinic, and they both have different things that they impact. So let's look at those very quickly. All right, you don't need to know all of the different muscarinic receptor sites. This is all I want you to know. I want you to know that muscarinic receptor sites, there are two main ones. There's the M2 receptor and there's the M3 receptor. The M2 receptor is a cardiac receptor. So when you target muscarinic receptor sites at the level of the cardiac system, you are going to cause a change in the heart rate. The other one that I want you to appreciate is that the M3 receptor has to do with sweat and sweat glands. So this is going to decrease heart rate, this is going to increase sweating, and you already knew this because earlier we said parasympathetic nervous system is feed or breed. We said acetylcholine is the neurotransmitter, and when the parasympathetic nervous system talks to the heart, it's a slowing action. It suppresses force, suppresses conduction velocity, and it suppresses heart rate. We also said when the parasympathetic nervous system stimulates sweat glands, it's going to cause sweating to take place. And I apologize here, I've made a mistake. Let me fix that real quickly. And this is not an increase in sweating. This is a decrease in sweating. All right, good. So parasympathetic nervous system, two different types of acetylcholine or cholinergic receptors. They are the muscarinic and nicotinic, and these guys change heart rate and they change sweating. The nicotinic receptor sites, they go to something called the neuromuscular junction. And the neuromuscular junction is where the nervous system interfaces with the muscular system. So wherever we have a nerve cell that touches a muscle cell to cause depolarization or to cause contraction of that muscle cell, that is called the neuromuscular junction. And the neuromuscular junction, in order for this nervous system to interface with the muscular system, it does so by way of a chemical neurotransmitter called acetylcholine, and the receptor site or the receiver in the neuromuscular junction on the muscle side of it is called a nicotinic receptor. All right, that's all you need to know for right now about that. All right, now let's look at the sympathetic nervous system. Sympathetic nervous system, I want you to think about adrenaline or epinephrine. All right, and adrenaline means, or adrenergic receptors are receptors that respond to adrenaline that come and that are primarily used in the sympathetic nervous system. And they, of course, have also their different receptor sites. So let's talk about their main ones. So in the sympathetic nervous system, adrenaline is the neurotransmitter. The receptor sites are called adrenergic receptor sites. And these receptors can be labeled very specifically, just like we did on this side. We're going to label them alpha receptors and beta receptors. We're also going to add the D, which is the dopamine receptor. All right, so let's look here first. Alpha receptors come in two flavors, alpha 1 and alpha 2. For our purposes, the only one we need to be familiar with is alpha 1. Alpha 1 receptors target smooth muscle cells in the peripheral vasculature, and they cause vasoconstriction. So, if you have an adrenergic drug, like adrenaline or adrenaline itself, it's going to target alpha 1 cells, and when you put that drug in the body, it's going to cause the smooth muscles of the peripheral vasculature to vasoconstrict, which means you're going to tentatively get an increase in blood pressure. That's the goal there. All right, so then we have to look at the next piece of this. And the next piece of this is, let's look at the beta receptors. And the beta receptors, there are several beta receptors. The only ones we need to know about are beta 1 and beta 2. Think about one heart. So beta 1 receptors are found in the heart. They increase heart rate, they increase the force, and they increase conduction velocity. Conduction velocity through the heart. And we'll examine this in greater detail when we get to cardiology. Beta 2 receptors, think about your lungs. You have two lungs. This is a terrible drawing of the lungs. You have two lungs. Maybe that will help. All right. No, it didn't. All right. You have two lungs. Beta 2 receptors are found in the bronchi, and they cause bronchodilation. But again, you already knew all this stuff because sympathetic nervous system is fight or flight. Fight or flight means I need to increase blood pressure. So vasoconstriction is going to do that for me. So it makes sense that an adrenergic receptor, if we have adrenergic receptors in the peripheral vasculature, it will cause those vessels to vasoconstrict. It just so happens that the name given to that particular receptor is called the alpha 1 receptor. It also makes sense, sympathetic nervous system, fight or flight, adrenaline, I got to increase heart rate, I got to increase force, I got to increase conduction velocity to be able to move all that oxygenated blood. And last but not least, I got to dilate the blood vessels. And the reason I dilate dilate the bronchi is so that I can move more air in and out of my lungs. These are all sympathetic responses. And last but not least, we have the D, or the dopamine or dopaminergic receptors. And these guys, as we mentioned, are renal smooth muscle targets. All right, so receptors, neurotransmitters, effects. What do you really need to know? You need to know that there are two divisions of the autonomic nervous system, parasympathetic and sympathetic nervous system. You need to know that in each of these divisions, there are neurotransmitters that primarily do all the work. We're talking about the neurotransmitters here that are post-ganglionic. They're at the end effector organ. So the neurotransmitter of the parasympathetic nervous system at the effector organ is acetylcholine. The neurotransmitter in the sympathetic nervous system end organ is adrenaline or norepinephrine. From there, we're going to look at specific receptor sites. The specific receptor sites that respond to acetylcholine post-ganglionic in the parasympathetic nervous system is acetylcholine. These are called cholinergic receptors. Cholinergic receptors can be broken down into two specific types of receptors. one of which is called muscarinic, the other is called nicotinic. Muscarinic receptor sites primarily impact cardiac, tissue, and sweat glands. The other cholinergic receptor is called a nicotinic receptor, and it primarily impacts the neuromuscular junction. This tells us about muscle contraction. All right, sympathetic nervous system. The receptors. We have alpha, beta, and dopaminergic receptors. The neurotransmitter in the postganglionic portion, the end effector neurotransmitter of the sympathetic nervous system, is adrenaline. Those receptors that respond to adrenaline are called adrenergic receptors. Alpha 1 receptors cause vasoconstriction. They're primarily found in the smooth muscle of peripheral vessels. Beta 1, primarily located in the heart, cause these increase in changes. Beta 2, there are two lungs, primarily found in lung tissue, but specifically in the bronchi and bronchioles of lung tissue, and they cause bronchodilation. Last but not least, we'll talk about dopaminergic. This is also part of the sympathetic nervous system, and these impact smooth muscle cells of the kidneys. All right, we're almost done. Bear with me. We're doing great. All right, so let's take a look at a couple things here. We need to understand a concept here, and the concept is the concept called resting membrane potential. And resting membrane potential is a concept that tells us about the potential or the stored energy that exists inside a cell, resting membrane potential, and the stored energy is that is available for us to do work with. So bear with me for a second. Let's take a look at a resting cell. All right, so here's a cell. And inside the cell, we've got a ton of stuff, but primarily we're going to look at the ions that are inside the cell. So inside the cell, we have potassium, we have chloride, and we have ATP. All right, chloride and potassium, both ions. Chloride's an anion, negative. Potassium's a cation, it's positive. And ATP, adenosine triphosphate, is the unit of currency for energy, and it is highly electronegative. All right, on the outside of the cell, we have a ton of sodium. Ton of sodium on the outside of the cell. All right, so here's how this works. If you were to take a little voltmeter, all right, you take a little voltmeter, and it's got a little arrow on here, and it's negative and positive, and you take a little voltmeter, and you put one of the probes of that voltmeter inside the cell, and you take another probe of that voltmeter and you rest it just outside the cell, and you look to see what the potential difference is, meaning what the electrical difference is from the inside of the cell to the outside of the cell. What you would come to realize and what you would measure is that the inside of the cell is about negative 70 millivolts. So the inside of the cell is negative compared to the outside of the cell. That means that there's a polarized system that exists here, meaning that there's a north and a south. There's a definite positive or negative. There's a definite black or white. So if you look at this here, if you look at the inside of the cell and you measure it, it is a little bit more negative than the surrounding area, and that little bit more negative is negative 70 millivolts. So the resting membrane potential is the amount of electrical difference or the potential stored energy that exists across this cell membrane, and the energy that exists across this cell membrane is negative 70 millivolts. That's how much energy exists there. And so at rest, when a cell is ready to go, it is said to be polarized. And a polarized cell just means that it has accomplished and achieved and maintained its resting membrane potential of negative 70 millivolts, negative on the inside compared to the outside. All right, let's move forward. So the next step is, all right, well, let's look at what happens if this cell membrane allows movement of ions to take place. So I'm just going to draw sodium, and I'm just going to draw potassium in here. And I'm going to say, now there are going to be these little channels, these little areas inside the cell membrane that allow for movement of potassium. They're called potassium channels. So potassium channels exist. And here we're going to put in another little embedded channel, and this channel is going to be a sodium channel, meaning that it specifically allows sodium to move through it, and this one specifically allows potassium to go through it. So when this cell is at rest, it's polarized, it's accomplished or achieved its resting membrane potential. If it is triggered, if there's a stimulus that touches this cell, that stimulus is going to trigger the opening of potassium and sodium channels. So when you open these channels, since there's a lot of potassium inside and none out here, we'll go back to diffusion. Remember, think of diffusion, what happens, and the movement of ions down their concentration gradient. Two things are going to happen here. Potassium is actually going to go through that potassium channel and it's going to escape. And sodium is going to do a couple things. It's going to actually travel through the sodium channel and it's going to enter the cell. And with these guys open, things are going to equalize such that the number of potassium inside the cell and outside the cell are going to try to equate. And the same thing is going to happen with sodium. And again, if you were to take your little volt probe now and you were to measure how much voltage exists across this cell membrane, if this is 0, you would find that it is 0 millivolts. So now we've gone from a polarized state to a depolarized state. So depolarization of cells is a fancy concept of illustrating the movement of ions from inside and surrounding cells such that they've equalized across the border. And if we were to then measure how much stored energy exists inside the cell, the answer would be 0, because there's equilibrium that has accomplished or achieved itself, and as a result, there is no stored energy, and this cell has done all the energy movement that it can, and now it's at rest. So polarized states are in a state of readiness to do work. Depolarized cells cannot do work. In fact, they can't respond to another stimulus. So if another stimulus, the same one that caused this thing to open these channels in the first place, hits this cell again, this cell can't possibly respond because it has no stored energy. It's depolarized, and it's in a state of something called refractoriness. And refractoriness just says, if I have a cell that is fully depolarized, it is refractory, it is unable to respond to another stimulus. All right, now, eventually, what will happen to this cell is the body will tell the cell, hey, you can't just hang out like that and be a lazy thug. You got to do something to get yourself back together. So in this case, what's going to happen is there's potassium out here, there's sodium in here, and we're now going to take this new little guy, and this new little guy is going to be here. It's going to be a sodium-potassium pump. And of course, the sodium-potassium pump is going to require energy because it's going to move ions where they don't want to go. It's going to use ATP. But this pump is essentially going to take this potassium, it's going to bind it, and it's going to dump that potassium back into the cell. It's going to take the sodium that's inside the cell and it's going to push it back outside the cell. So now potassium's back inside, sodium's back outside, and if you were to take that little voltmeter again and you were to measure the potential energy, here's 0, and the potential energy that exists between the outside and the inside of the cell, you would find that inside the cell you were at negative 70 millivolts. This is called repolarized. Repolarized. And the cycle repeats itself. So essentially what you have here is you've got a cycle that takes a cell at rest with stored energy, kind of like a battery. You plug the battery in until you completely deplete it. This is called depolarization. When you're depolarized, eventually the body will take it and say, hey, we got to fix it again. It will repolarize the cell. It will reestablish the resting membrane potential, It will restore the cell to being ready to do work again or to be stimulated again and to respond to that stimulus. And that cycle can continue over and over and over and over again. All right, so now why in heaven's name have we just talked about all this physiology? And the answer is simple, because we are going to impact all of these things either with different disease processes that the patient has, or more importantly, with certain drugs that we give the patient or certain therapies, we can actually directly target this stuff. So let's take a look at how we're going to do that. All right, so let's talk about nerve impulse propagation. So remember, we just talked about depolarization and repolarization. Now we're going to apply that theory, that concept, to something actually that we can use. All right, so here is an axon, meaning that over here we have a nerve cell, and it's got all these little dendritic endings, all these little endings that can receive stimulation. All right, all these little guys here. This is a cell body. Here's the big nucleus inside that. All right, so here's what's going to happen. Remember that we said outside of the cell, so outside of this axon, here's an axon. Here's the cell body of the nerve cell. All right, and then over here we're going to have an organ. All right, so here's the axon. This is the inside of the axon. This is the outside. And as you'll remember, tons of sodium on the outside, lots of sodium on the outside, lots of sodium on the outside, lots of sodium. All this sodium out here. It's all around this axon. It is essentially bathing in a salt bath. All right, so there's all this sodium. And inside, it's negative. So we're going to just represent this with negatives on the inside of this axon. So if you were to look at the cell membrane here, this cell membrane is the barrier that separates the outside and the inside of these two systems. And outside it's predominantly positive, and inside it is predominantly negative. Now, just for the purposes of helping myself show you here, what I'd like to do is I'm going to take out all of these sodiums and I'm going to replace them with just plus signs so that you can see what's going on here. But I want you to remember that all of those little plus signs that I'm going to draw here are actually sodium. So this is all sodium, 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 sodium. So there's all this sodium right outside here, and this is an axon. All right, so here's what happens. What happens is there's a trigger, and we'll talk more about what the trigger can be, and that trigger touches this little guy here. And the trigger is brought into the cell body, and the cell body does some chemical manipulation of this trigger, and essentially what it does is it takes this little channel, a little sodium channel that's here, and it opens it. So this trigger was chemically converted by the cell body. The cell body did something to change the permeability of sodium channels in the axon itself. It opens the sodium channel, and when it opens that sodium channel, sodium rushes in, making the inside a little more positive, and it continues to do that. So the inside of the cell now becomes positive. When you change the polarity of the cell, it goes from a polarized state, when it was negative inside, to now a depolarized state. When you depolarize a cell, it causes an impulse to be formed. So there's now an electrical impulse, which is actually nothing more than sodium just moving open all the way down the axon. And that impulse propagates all the way down this axon until it reaches the end of the axon where there's an organ. All right, so nerve impulse propagation solely relies on sodium influx, the movement of sodium into the axon. And sodium influx into the axon propagates the nerve impulse, and it causes it to get all the way to the end of the axon, where it is chemically converted, and that chemical conversion is acetylcholine for our purposes here. And the acetylcholine goes to the organ where there are cholinergic receptors. And if there's a cholinergic receptor on an organ, it causes the organ to do something. It might be to release something, or to secrete something, or to do something. All right, so sodium influx. This is a really, really important concept because we're going to talk a lot about what we can do to prevent this. So, in fact, we can prevent the propagation of nerve impulses by preventing sodium channels from opening. And we can do that with drugs such as sodium channel blockers. So everything we do in medicine with respect to pharmacology is going to impact some sort of ion channel. It could be a sodium channel, could be a potassium channel, could be a calcium channel, could be a chloride channel, could be all sorts of different things. All of these are going to change the way the body responds or propagates an impulse or causes a change to one of the sites. All right, so nerve impulse propagation heavily relies on sodium, and it causes propagation all the way down to the organ where it is converted chemically to a neurotransmitter. If it's acetylcholine, it binds to a cholinergic receptor, and then it causes the end organ to do something or to release something. All right, so I think that is pretty heavy on this stuff. So let's give this a little bit of a break. We'll come back and talk more about this with some very specifics about the different drugs. Stay tuned.